Welcome to the presentation of biophysically grounded mean field models of neural networks under electrical stimulation, which was published in PLOS Computational Biology. First, I want to talk about the motivation of this work. Brain stimulation, or more specifically transcranial electrical stimulation, is a widely used tool known to be able to modulate ongoing brain activity and has many different clinical applications. A comprehensive modeling approach of this would be to simulate a whole brain model and add an electric external stimulus to the nodes of the brain network. However, it is not yet well understood how electrical stimulation affects the dynamics of the underlying neural populations themselves. An appropriate in vitro setup to investigate this would be, for example, a cortical slice surrounded by an electric field. In these frequency spectrograms, you can see how a direct and an alternating external field can turn on and off oscillations and modulate them in a neural slice preparation. In this work, we want to investigate the mechanisms of how external stimulation affects the dynamics of a population model that represents a piece of neural tissue. The presented mean field model is derived from a large network of excitatory and inhibitory adaptive exponential integrate and fire neurons. While the dimensionality of the original description is vast and therefore very cumbersome to analyze and to simulate, the resulting mean field model has only 10 ordinary differential equations that represent the macroscopic variables of the system. In order to derive this model, we make the following assumptions. We assume the number of neurons to be infinitely large, all units to be equal, the connectivity to be sparse and random, and make use of the diffusion and the adiabatic approximations. This results in a much simplified and rate model of the system that depends on the mean and the standard deviations of the membrane currents. Let's discover what this model is capable of doing. Here we see a bifurcation diagram of the population model. The x and the y-axis represent the mean background inputs to the excitatory and the inhibitory subpopulations. We can see different attractors, such as a steady state with a high activity upstate and a low activity downstate, a bistable region with a green contour where both of these states coexist, and a limit cycle region with white contours where the activity alternates between the excitatory and the inhibitory populations. When we compute the bifurcation diagrams of the underlying neural network, we can see that, indeed, the state space looks very similar, indicating that the mean field model is able to represent the original model fairly well. All attractors are present and the bifurcations take place at similar locations. In the next step, we turn on a somatic spike frequency and a subthreshold adaptation mechanism. In a single AdEx neuron, this leads to a slowing down of the spiking in response to a constant stimulus. The effects on the whole population activity, however, depends on the location in state space. The most important effect of adaptation is that it transforms the bistable region into a new limit cycle in which the activity oscillates very slowly due to excitation and adaptation feedback. Here we can see the time series of both models when we parameterize the system to be in the limit cycles. In the upper row, you can see a fast oscillation in a low gamma range between E and I. In the lower row, we observe a very slow oscillation that is generated by excitation and adaptation. The bifurcation diagrams help us to understand what happens when the system is subject to external stimulation. In this picture, an external electric current or field can be thought of as a movement in the x-axis, considering that electric field stimulation primarily affects excitatory pyramidal neurons. We can then understand how a constant stimulus is able to cause attractor switching and thus turn on and off oscillations. We can also directly measure the bistable region by applying a decaying stimulus. When we apply an oscillatory stimulus, we can observe phase locking, frequency entrainment, and driven oscillations. In this last slide, we explore the response to non-stationary input in more detail. Here on the left, you can see the frequency spectrum of the neural activity plotted against the changing stimulus frequency. We can see how the external stimulus is able to frequency entrain the system, meaning that the external stimulus can change the frequency of the neural oscillation. This happens at equivalent electric field strengths of about 5 volts per meter or stronger. Here on the right, we measure phase locking as a function of stimulus frequency and amplitude. The diagrams show Arnold tongues where the stimulus is able to dictate the phase of the ongoing neural oscillation. This effect is visible for much weaker electric fields on the order of 1 volt per meter. Both effects are well captured by the mean field model and confirmed by simulations of large neural networks, further demonstrating that the applicability of mean field models extends to the non-stationary case. To summarize, we have seen that mean field approximation works and that it also works in a non-stationary case. We've seen that electric field effects depend critically on the state of the system, that DC inputs can cause attractor switching, and that AC inputs can frequency and phase-entrain ongoing oscillations. Furthermore, we have calculated and predicted electric field strengths at which these observations are to be expected, which is well in line with experimentally reported values. With that, I thank you for your attention.